Good morning, student. We will continue topic protein metabolism, competition number 5.4. So today's subtopic is metabolic pathways of sulfur containing amino acids like methionine, cysteine and cysteine. At the same time, we are also studying their role in biosynthesis of various specialized biomolecules and metabolic disorders associated with these amino acids. First, methionine, it is essential amino acids. It is glucogenic amino acid. That means on catabolism it forms glucose. Cysteine, it is non-essential amino acid because it is synthesized from methionine. It is also a glucogenic amino acid. Cysteine. Now cysteine is nothing but it is a dimer of cysteine joined together by disulfide bond. So this is the structure of cysteine, where you see two cysteine molecules combines together, their SH group forms a disulfide bond in cysteine. Now first about methionine. So methionine and active methionine. S-adenosyl methionine is also called as active methionine. Why? Because it participates in various transmethylation reactions. So on the left side I have shown the structure of methionine. Now this methionine combines with adenosyl group of ATP. ATP is made up of adeno, ribose sugar, adenine and 3-phosphate group. So adenine plus ribose sugar, they form adenosyl group. So that adenosyl group combines with the sulfur group S atom of methionine to form S-adenosyl methionine. The enzyme is methionine adenosyl transferase. So due to binding of adenosyl group at sulfur atom, the attachment between sulfur atom and methyl group becomes weak. Therefore, this methyl group can be easily detached from S-adenosyl methionine, which is very important in transmethylation reactions. Next is S-adenosyl methionine cycle or SAM cycle. Short form of S-adenosyl methionine is SAM. Now here methionine combines with ATP, where adenosyl group of ATP combines with sulfur atom of methionine to form S-adenosyl methionine, due to which the methyl group is easily detached from S-adenosyl methionine, and this methyl group can be added to any acceptor to form methyl plus acceptor. After removal of methyl group from S-adenosyl methionine, it is converted into S-adenosyl homocysteine, SAH. Right. Now later on in the third reaction, the adenosyl group is also removed from this S-adenosyl homocysteine and it becomes just homocysteine. Now this homocysteine, when combines with methyl tetrahydrofolate, this methyl tetrahydrofolate is a carrier of one carbon compounds where it carries methyl group. Now this methyl group is donated to the homocysteine by methyl tetrahydrofolate with the help of vitamin B12 or cobalamin, and this methyl group then binds with sulfur atom to regenerate a methionine. So this cycle starts from methionine and ends with methionine. So therefore it is called as SAM cycle. This is the actual SAM cycle that you have to draw in the exam. So here the same is written. Methionine combines with ATP, where 3-phosphate groups are removed and adenosyl group is added to methionine to form S-adenosyl methionine. This S-adenosyl methionine donate methyl group to acceptor to form methylated product and get converted into S-adenosyl homocysteine, SAH. Later on, the S-adenosyl homocysteine donate adenosyl group to form homocysteine. Now this homocysteine then combines with a methyl group which is obtained from methyl cobalamin. but actually this methyl group is donated by methyl tetrahydrofolate, and cobalamin is just a mediator. So methyl tetrahydrofolate donate first methyl group to cobalamin to form methyl cobalamin. This methyl cobalamin then donate methyl group to homocysteine to regenerate methionine. So in this way the SAM cycle takes place. The reaction required for the first step, where methionine is converted to S-adenosyl homocysteine, is methionine adenosyl transferase. For the second step, the enzyme is methyl transferase, where S-adenosyl homocysteine donate methyl group and get converted into S-adenosyl homocysteine. For the third step, where adenosyl group is removed from S-adenosyl homocysteine, or you can say S-adenosyl homocysteine is broken down into two compounds, homocysteine and adenosyl. The enzyme is adenosyl homocysteinase. And the fourth step, when homocysteine accept a methyl group, the enzyme is homocysteine methyl transferase. And for the fifth step, where methyl tetrahydrofolate donates methyl group to cobalamin, the enzyme is 5-methyl tetrahydrofolate homocysteine methyl transferase. So this is a SAM cycle, which may be asked as a short note. Next is transmethylation reactions. Transmethylation reactions are the reactions where active methionine, that is S-adenosyl methionine, donate methyl group to various compounds, and in that process S-adenosyl homocysteine is formed from S-adenosyl methionine. So this is the transmethylation. These are transmethylation reactions. First, I have shown methyl acceptor. I will give you the list of methyl acceptor. It accept methyl group from S-adenosyl homocysteine and get converted to methylated product. I also mentioned the methylated products. Then S-adenosyl homocysteine is converted to S-adenosyl homocysteine. So these are the examples of transmethylation reactions. First is guanidoacetate. Guanidoacetate here act as a methyl acceptor, which accept methyl group from SAM and get converted into methylated product called creatine. So SAM is converted to SH. Second methyl acceptor is nicotinamide, which accept methyl group and forms a methylated product called N-methyl nicotinamide. Third example is norepinephrine, accept methyl group and form a methylated product called epinephrine. In the next reaction, epinephrine itself act as a methyl acceptor and forms a methylated product called metaepinephrine. Next methyl acceptor is acetylserotonin, which forms melatonin as a methylated product. Ethylamine combines with methyl group of SAM to form methylated product called choline. At the same time, carnosine also act as a methyl acceptor from SAM to form a methylated product, and serine. Histidine also combines with methyl group of SAM to form methyl histidine. Serine on methylation forms choline. Nicotinamide on methylation forms N-methyl nicotinamide. Lysine forms methyl lysine. tRNA form methylated tRNA. So these are the list of methyl acceptor and methylated product in transmethylation reactions. After completing SAM cycle and transmethylation reaction, we will start metabolism of cysteine. Cysteine is a non-essential amino acid. It is synthesized from methionine. So here is the SAM cycle again. In the SAM cycle, I have told you that methionine is converted into S-adenosyl methionine by accepting adenosyl group from ATP. That S-adenosyl methionine donate methyl group to acceptor and form S-adenosyl homocysteine. This S-adenosyl homocysteine donates adenosyl group to form homocysteine. In the SAM cycle, I have told you that this homocysteine is converted into methionine with the help of two vitamins, that is folic acid, that is coenzyme of folic acid, that is tetrahydrofolate, and vitamin B12. But this homocysteine, some part, is also converted into cystathionine by combining with serine. So this reaction takes place according to the need of the cell. Means some part of homocysteine is remethylated to methionine, or some part of homocysteine combines with serine to form cystathionine. The enzyme is cystathionine synthase. Here dehydration takes place. Then cystathionine is broken down into two compounds, cysteine and homoserine, with the help of enzyme cystathionase. This cystathionase is a hydrolase enzyme. It breaks the cystathionine with the help of water molecule. Here the coenzyme is vitamin B6 or coenzyme of vitamin B6, that is pyridoxal phosphate, is required. After studying the synthesis of cysteine, we will see catabolism of cysteine. Catabolism of cysteine takes place by two processes, transamination and decarboxylation. Transamination process, in which cysteine undergoes deamination to form a compound called beta mercaptopyruvate in the presence of pyridoxal phosphate. It is not transamination, it is not deamination, it is a transamination. Here the amino group is accepted by alpha ketoglutarate to form glutamic acid. This beta mercaptopyruvate is formed, and this beta mercaptopyruvate, when releases its sulphur group in the form of hydrogen sulfide, it forms pyruvate. This is the first process. It is not deamination, it is a transamination. Now H2S, which is formed here, is a very important compound. Now this H2S or hydrogen sulfide get oxidized to form sulfide, which further oxidize is to sulfide, and after one more oxidation it forms sulfate. So hydrogen sulfide on oxidation forms sulfate. Now this sulfate has two fate. Either it is excreted into the urine, or this sulfate may be converted into PAPS. Long form is phosphoadenosyl phosphosulfate. Now this phosphoadenosyl phosphosulfate is called as active sulfur. Now this is phosphoadenosyl phosphosulfate or active sulfur. Now see here what happens. The sulfate, which is formed from H2S, combines with two ATPs. One ATP is broken down into AMP and two phosphate bonds are removed. This AMP directly combines with sulfate, and another ATP is hydrolyzed to ADP and inorganic phosphate. Here ADP is removed, and the inorganic phosphate of second ATP combines with the third carbon atom of AMP to synthesize a compound called as phosphoadenosyl phosphosulfate or active sulfur. What is the role of active sulfur or phosphoadenosyl phosphosulfate? So it is used for synthesis of mucopolysaccharides. Mucopolysaccharides, all of us know, glycosaminoglycans. In carbohydrate chemistry you have studied. Then this is also used for synthesis of glycolipids and sulfolipids, where it donates sulfur group in the synthesis of all these three compounds. Next catabolism reaction of cysteine is decarboxylation. Cysteine on decarboxylation forms beta mercaptoethanolamine. So here the structure is shown. The CO2 group is removed, therefore cysteine is converted to beta mercaptoethanolamine. Later on this beta mercaptoethanolamine is converted into coenzyme A, that we will see later on. So by these two ways cysteine is catabolized in the body. First is transamination and this is decarboxylation. Now this is important. Biologically important substances synthesized from cysteine in the body. First is synthesis of glutathione, that we will see. Now what is glutathione? Glutathione is a tripeptide made up of cysteine, glycine and glutamic acid. First, glutamic acid or gamma glutamic acid. Glutamic acid and gamma glutamic acid is same. Here glutamic acid is called as gamma glutamic acid because glutamic acid have extra carboxyl group, and that extra carboxyl group is attached to the gamma carbon of glutamic acid. So gamma glutamic acid combines with cysteine to form gamma glutamyl cysteine. The enzyme is glutamyl cysteine synthetase. Here, for combination of these two amino acid, energy is required, and that is supplied by ATP. Therefore, enzyme is synthetase. In the next step, gamma glutamyl cysteine combines with the third amino acid glycine. Again, here for joining this glycine to the substrate, energy is required, which is supplied by ATP, to form a compound called as gamma glutamyl cysteanyl glycine, which is also called as glutathione, and the name of enzyme is glutathione synthetase. So in this way, glutathione is synthesized where cysteine plays an important role. Now this glutathione is a very important compound in our body, so there may be chances that there is a short note on glutathione. So we will study glutathione in details, or role of glutathione. This may be MCQ question. First, to study the role of glutathione, you should know the mechanism of action of glutathione. Glutathione actually acts as a reducing agent. That means it supplies hydrogen to the substrate, or it will add hydrogen to the substrate and leads to reduction of that substrate. Sometimes it will add electron to the substance, leading to reduction. Addition of hydrogen or electron, both are reduction process. So in this left side I have written 2GSH. H is the hydrogen atom which is present along with glutathione. So this glutathione is called as reduced glutathione. This reduced glutathione, when donated two hydrogen atom to the substance, it forms oxidized glutathione, which is represented by GS. So it is oxidized glutathione. So simple role of glutathione is it acts as a reducing agent. Now we will see the examples and the importance of the reaction where glutathione acts as a reducing agent. First is maintenance of RBC membrane integrity. Now in the RBCs, superoxide anion is continuously formed. Now this superoxide anion is converted into hydrogen peroxide by an enzyme superoxide dismutase. Now this hydrogen peroxide is a reactive oxygen species because it is formed by incomplete reduction of oxygen molecule. Now this hydrogen peroxide is harmful for RBCs. This hydrogen peroxide is converted into water molecule by adding two more hydrogen atoms from glutathione, reduced glutathione, by an enzyme glutathione peroxidase. So in this way, the hydrogen peroxide is removed from the RBCs. Here both the enzymes superoxide dismutase as well as glutathione peroxidase, they are called as antioxidants because they helps to remove the oxidant substances like hydrogen peroxide and hydrogen peroxide from the RBCs. These two substances are harmful for the RBCs. If the level of these substances in the RBC increases, it will lead to hemolysis. So in this way, glutathione plays an important role in RBC membrane integrity. Next is glutathione act as a coenzyme in various reactions. First reaction is conversion of mallyl acetoacetate to fumaryl acetoacetate. So this is the one of the reaction in tyrosine catabolism pathway that we have studied in the last lecture. So this is the tyrosine catabolism pathway. If you see here, conversion of 4-mallyl acetoacetate is converted to fumaryl acetoacetate by an enzyme isomerase, where glutathione acts as a coenzyme. Second is conversion of citric acid to taurine. In this reaction also, glutathione acts as a coenzyme. So this is the reaction. Cysteine first get converted, oxidized to citric acid where SH group of cysteine is converted into SO3H. Now this cystic acid undergo decarboxylation reaction to form taurine, where glutathione acts as a coenzyme. Later on, this taurine combines with cholic acid to form taurocholic acid, which is a bile acid. And third reaction is conversion of iodine molecule into hydrogen iodide. In our diet, iodine is present in iodine molecular form, but this molecular oxygen cannot be utilized by the thyroid gland. Therefore, reduced glutathione provides hydrogen atom to this molecular oxygen, molecular iodine, and get reduced to hydrogen iodide, which is utilizable form. Next role of glutathione is conversion of methemoglobin to hemoglobin. So the difference between methemoglobin and hemoglobin is, in methemoglobin iron is in ferric state, whereas in hemoglobin iron is in ferrous state. Ferric iron is pentavalent, ferrous iron is hexavalent. So this hexavalent ferrous iron has 6 valencies. By 5 valencies it combines with globin chain, and with 6th valency it combines with oxygen molecule. Whereas in methemoglobin ferric iron is present, which is pentavalent. So it has 5 valencies and it only combines with globin chain. So it does not combines with oxygen molecule. Therefore, methemoglobin is not for transport of oxygen because it does not combines with oxygen. Now, in our body, continuously this methemoglobin is formed. Why? Because iron want a stable state. The stable state of iron is ferric form. So iron readily get oxidized to achieve ferric state, and hemoglobin is converted into methemoglobin. So there is a constant formation of methemoglobin from hemoglobin. So this methemoglobin should be converted back to hemoglobin. So here reduced glutathione provides electrons to the ferric iron of methemoglobin and convert it into ferrous form. In this way it converts methemoglobin into hemoglobin. Next is detoxification. What is detoxification? It is conversion of toxic substances into less toxic substances. Toxic substances are water insoluble or less soluble in water, whereas non-toxic substances are more soluble in water and therefore they can be easily removed from the body. Now here glutathione donates cysteine to the toxic substance in the conjugation reaction. Conjugation means joining reaction. When this cysteine joins with a toxic substance X, the solubility of this substance increases, and therefore the compound X, which is less soluble in water, now becomes more soluble in water. Therefore it is less toxic for the body, and body can easily remove from the body can easily remove it outside. Now this is the mechanism. Glutathione, first glutamine group is removed from glutathione, then cysteine glycine is remaining. Now this cysteine glycine donates citric acid to the toxic compound, and toxic compound get converted into non-toxic compound because it becomes more soluble. So this is the detoxification process. Now glutathione donates cysteine group in conjugation. This type of reaction is called as conjugation reaction for the following substances: organophosphorus compounds, halogenated compounds, heavy metals, and drugs. So these are toxic substances. When these substances combines with cysteine, they becomes more soluble and get easily excreted from the body. Now next important compound obtained from cysteine is taurine. After glutathione, taurine. I have already told you this. Cysteine on oxidation forms citric acid, where SH group is converted into SO3H group. Now citric acid on decarboxylation forms taurine, where glutathione acts as a coenzyme. Now this taurine combines with choline-CoA. Choline-CoA means it is a combination of cholic acid and coenzyme A. So this choline-CoA combines with taurine, where the choline group or cholic acid is transferred to taurine to form taurocholate or taurocholic acid, which is the one of the bile acid, and coenzyme A is removed. Coenzyme A is always written as CoA-SH. Why it is SH is written? Because SH is the active group present in coenzyme A. To show that active group, coenzyme A, free coenzyme A is always written as CoA-SH. But when this coenzyme A combines with any compound, then it is not necessary to write down SH. See, on the left side, just choline-CoA is written, where SH group is not shown. But when coenzyme is free, then separately we have to write down SH group. Next compound obtained from cysteine is glucose. Cysteine undergoes transamination reaction in the presence of pyridoxal phosphate to form beta mercaptopyruvate, and this beta mercaptopyruvate, when hydrogen sulfide group is removed, forms pyruvate that we have studied, and this pyruvate by gluconeogenesis get converted into glucose. So cysteine is a glucogenic amino acid. Next is maintenance of protein structure. Cysteine has SH group, sulfhydryl group. When two cysteine molecules came close to each other, their sulfhydryl group combines to form a disulfide bond, SS bond, and in this way they helps to maintain the structure of a polypeptide chains or proteins. Next important role of cysteine is synthesis of coenzyme A. I have told you cysteine on decarboxylation form beta mercaptoethylamine, and this later on get converted into coenzyme A. This coenzyme A you have seen in various combines with various compounds like acetyl-CoA, succinyl-CoA. Just now we have studied choline-CoA. After completing the important compound obtained from cysteine, we will study disorders of sulfur containing amino acids. So these are the disorders of sulfur containing amino acids: cystinuria, cystinosis, hypermethylaminemias, homocystinemia, and homocysteinurias, cystathionuria. We will study one by one. Cystinuria, as the name suggests, excretion of cysteine in urine is called as cystinuria. It is a autosomal recessive disorder. In this disorder, not only cysteine but 3 more amino acids like ornithine, arginine, and lysine are excreted into the urine, but name is cystinuria. In this disorder, the basic defect is reabsorption of amino acid from renal tubules. All of us know that free amino acids are easily filtered through the glomerulus, but later on they can be reabsorbed by proximal columnated tubules. But in cystinuria, the transporters which transport these 4 amino acids from filtrate to the body are absent. Therefore, reabsorption of these 4 amino acids is hampered and they get excreted into the urine, and it is called as cystinuria. Next is cystinosis. In this case, there is accumulation of cysteine crystals in the lysosomes of various organs like liver, spleen, bone marrow, WBC, kidneys, cornea, and lymph nodes. So all of us know that lysosomes are present inside the cell, which leads to hydrolysis of unwanted substances inside the cell. In our cell, there is constant breakdown of proteins and synthesis of new proteins from the amino acids which are generated from the breakdown of previous proteins. So this is called as protein turnover. So normally what happens, the proteins which has to catabolize, they enter into the lysosome. The lysosome break this protein into amino acids. Then the amino acids from lysosomes enter back into the cytoplasma, and from these amino acids new proteins are synthesized by the cell. In cystinosis, the lysosome will break the protein into amino acids. All the amino acids will enter back into cytoplasma except cysteine, because here again the transporters which require for the transport of cysteine from lysosome to cytoplasma are absent. Therefore cysteines are not transported to the cytoplasma, and this cysteine get deposited into the lysosomes, leading to cysteine crystals. When the concentration of this cysteine crystal increases, they will damage the lysosomes. So this is cystinosis. The basic defect is transport of cysteine from lysosome to cytoplasm. Next is hypermethyonemias. It is due to two reasons, either due to impaired utilization of methionine or excessive remethylation of homocysteine. To understand this, you have to see this pathway. See, in this case homocysteine is there. If this homocysteine is methylated more, then more methionine is formed. Actually this homocysteine has two roles. Either it is converted into cystathionine or it may be converted into methionine. But excessive remethylation of homocysteine causes synthesis of more and more methionine. So this is the one reason. Second is decreased utilization of methionine for transmethylation reaction. So methionine, I have told you, get converted into S-adenosyl methionine. If that reaction is hampered, again the methionine level is increased. So hypermethionemias are due to two reasons, either due to decreased utilization of methionine in SAM cycle or excessive remethylation of homocysteine to methionine. Next disorder is homocystinemia and homocystinuria. Homocystinemia means increase in the homocysteine level of blood, and homocystinuria means excretion of homocysteine in the urine. It may be due to congenital or acquired. Congenital are usually due to deficiency of various enzymes. So first we will see congenital homocystinemia and homocystinuria. So in this case the enzyme beta cystathionine synthase is deficient. Now this enzyme, if you see the cycle, convert homocysteine and serine into cystathionine. So if this enzyme is deficient, cystathionine synthase or beta cystathionine synthase, the homocysteine is not converted into cystathionine. So the level of homocysteine remains in the blood, increase, leading to homocystinemia and homocystinuria. So this is congenital due to deficiency of enzyme. Now this is also a congenital type of homocystinemia and homocystinuria. It is due to deficiency of one more enzyme called N5-N10 methylene tetrahydrofolate reductase. Now what is the role of this enzyme? This N5-N10 methylene tetrahydrofolate reductase convert the N5-N10 methylene tetrahydrofolate into N5-methyl tetrahydrofolate. N5-N10 or N5, that are the attachment of this methylene or methyl group to the tetrahydrofolate. So we have seen in the cycle methyl tetrahydrofolate or N5-methyl tetrahydrofolate act as a methyl donor in SAM cycle where homocysteine is converted into methionine. But this methyl tetrahydrofolate is formed from methylene tetrahydrofolate. And for the conversion of this methylene tetrahydrofolate to methyl tetrahydrofolate, the enzyme methylene tetrahydrofolate reductase is required. If this enzyme is deficient, then there is decreased synthesis of methyl tetrahydrofolate. And when methyl tetrahydrofolate deficiency is there, then conversion of homocysteine to methionine is hampered and the homocysteine level in the blood increases, leading to homocysteinemia and homocystinuria. Now coming to acquired homocysteinemia and homocysteinurias. Congenital means present since birth, acquired means they are acquired during the lifetime. So causes are nutritional deficiency like deficiency of cobalamin, folic acid, pyridoxine, or it may be drug induced like folate antagonist, pyridoxine antagonist, cobalamin antagonist, or it may be metabolic cause, chronic renal disease and hypothyroidism. First is nutritional deficiency. 3 vitamins are very essential in the metabolism of cysteine. These are folic acid, which provide methyl group in the form of methyl tetrahydrofolate, then cobalamin, that is vitamin B12, and B6, that is pyridoxine. So folic acid and vitamin B12 are required for conversion of homocysteine to methionine. If these 2 vitamins are deficient, homocysteine is not converted into methionine, therefore the level of homocysteine in the blood increases. Now this pyridoxine or vitamin B6 is required for conversion of homocysteine to cysteine. If you see here, homocysteine first converted into cystathionine, and that cystathionine is broken down into cysteine and homoserine, where vitamin B6 or pyridoxine is required. So if these 3 amino acids, if these 3 vitamins are deficient, then homocysteine is not properly utilized and its level in the blood increases, leading to homocysteinemia and homocystinuria, which is a acquired type. Next is antagonist drug for these vitamins: folate antagonist, pyridoxine antagonist and cobalamin antagonist. This drug will inhibit the above vitamins and therefore again homocysteine is not utilized. In metabolic cause like chronic renal failure and hypothyroidism, the enzyme required for the cysteine or methionine metabolism are not synthesized in proper quantity in the body. Therefore, homocysteine is not utilized and its level in the blood increases. Next disorder is cystathionuria. Again, it is also due to congenital and acquired. Congenital is due to deficiency of enzyme cystathionase and acquired is due to deficiency of vitamin B6. Here is the cycle all of us know, where cystathionine is broken down into cysteine and homoserine with the help of enzyme cystathionase, and coenzyme is vitamin B6. If the enzyme cystathionase is deficient, then cystathionine is not broken down into cysteine and homoserine. The level of cystathionine increases, leading to, and that cystathionine is get excreted into the urine, leading to cystathionuria. And acquired is due to deficiency of pyridoxal phosphate or vitamin B6. Pyridoxal phosphate is the coenzyme of pyridoxine. So in both these cases, cystathionine is not catabolized and it excreted into the urine. So this is about the metabolism of sulfur containing amino acid, important products obtained from cysteine, and various metabolic disorders associated with sulfur containing amino acid. Thank you.